Bueno, buenas tardes. Ahora sí nos dieron el luz verde para continuar, iniciar nuestro último seminario del año. Qué bueno que están acá. Y me da mucho gusto presentarles hoy a nuestro invitado, el doctor Matt Barbour. Matt nació en San Diego, California. Tiene una maestría por la Universidad de San Diego, estatal. En el 2012 lo obtuvo en el laboratorio del doctor Rulon Clark. Después se fue a hacer un doctorado a Vancouver, a Canadá, donde obtuvo su doctorado en la Universidad de British Columbia en el 16, con el doctor Greg Crossscale. Y después se fue a hacer una estancia postdoctoral a la Universidad de Zurich con el doctor Jordi Bascomtet, y recientemente, se acaba de cambiar de Suiza a Quebec. Va a iniciar su nuevo laboratorio. Es realmente joven y está empezando su carrera científica con bastante éxito de su postdoc. Matt combina la genómica y las interacciones interespecie en los diferentes ecosistemas para tratar de entender cómo esos cambios en los ecosistemas o cómo se mantienen los genes y la expresión génica entre las especies que interactúan en un mundo que está cambiando constantemente. Y hace investigación de campo, usa también investigación en laboratorio, pero una de las cosas que hace mucho son modelados matemáticos y de interacciones entre especies para tratar de contestar las preguntas interesantes que está generando en su laboratorio. Entonces integra la genómica de las interacciones entre las redes entre herbívoros y plantas para tratar de entender cómo están coevolucionando en este mundo. Entonces le doy muchas gracias a Matt por haber aceptado la invitación y platicarnos de esta, creo que es una nueva área de investigación que va a ser muy relevante en el futuro. Adelante, Matt, gracias.
All right, gracias Gustavo for that introduction, and gracias a todos for coming to this seminar, and thank you also to the Institute for inviting me to give this seminar. It's been really great meeting some of you so far, and I'm looking forward to meeting with many of you later. So I'm a community ecologist that's fascinated by evolution, and my work really lies at the intersection of these two fields. I spend a lot of time thinking about how evolutionary processes influence interactions between species and how those interactions affect the structure and dynamics of ecological communities. But I'm also interested in how changes in ecological communities can feed back to try to drive genetic and phenotypic change within populations. And so I'll start my talk by showing you why my research program is working at the intersection of these fields of community ecology and evolutionary biology. I'll then dive into the details of the major research projects that I've led and that I'm leading right now. And at the end of my talk, I'll leave plenty of time for questions where hopefully we can discuss how some of this work that I'm doing might intersect with the interests of with the interest in biotechnology here at the Institute. So I want to start my talk by showing you a painting that I feel beautifully illustrates what I think is a grand challenge that we face today in biology. And that's understanding how biological processes scale from genes to complex ecosystems. So in the lower part of this painting is a unicellular green algae. The DNA of this algae encode information that determine the structure and function of its chloroplasts that you see here in green. Now, these chloroplasts, of course, serve an important function as they harvest photons of light from the sun, which you can see as these tiny red dots, and through the process of photosynthesis, turn the sunlight into chemical energy for the algae, which also oxygenates this freshwater ecosystem as well as the atmosphere. Now, what I also want you to notice is that this algae is at the base of a food web where it's a resource for this Daphnia. This Daphnia is in turn a prey item for this fish, and this fish is preyed upon by this heron. So what I feel this painting beautifully illustrates is that gene to ecosystem processes sustain life on Earth. And so I think it's critical that we understand these linkages across biological scales if we want to preserve biodiversity in the face of environmental change. Now, over the past 30 years, we've made substantial progress in understanding these cross-scale linkages. And much of this body of work was nicely summarized by Andrew Hendry in his book on eco-evolutionary dynamics. So the things that we know are that we know that genetic variation influences phenotypic variation within species. Okay, the genes that you have determine your phenotype. We also know these phenotypes affect population dynamics as well as the structure and dynamics of ecological communities, but that also changes in communities and in populations can feed back to drive evolutionary change in phenotypes as well as heritable genetic changes. So one of the things we've realized is that there are these linkages across scales. But although we've done a really good job of documenting the potential for these cross-scale effects, I'd say what we're still very constrained in our ability to do is to predict how changes at one level of organization cascade to affect other levels. So for example, being able to understand how genetic variation within species affects ecological communities, we're still really far away from being able to do that. And I think that's because most research that has studied these cross-scale linkages ignores the interconnected nature of biodiversity at these different scales. So just as a bit of an example, I want to show you what many of you might be familiar with this example, but this is my favorite example. It comes from Bob Payne's classic experiment, where he showed that if you remove sea stars, this top predator species, in this intertidal ecosystem, that it alters the structure of interactions between species in such a way that just results in a few species outcompeting the others for limited space in the intertidal. So what this experiment showed is that ecological communities, they aren't simply a collection of species, but that they're highly structured by interactions, and it's the strength and organization of these interactions that determine how communities respond to environmental change. So if we want to predict how biodiversity is going to be reorganized by environmental change, if we want to predict how changes at one of these levels of organization is going to cascade to affect others, we need to account for this interconnected nature of biodiversity. And I think by doing this, we can start to answer a fundamentally important question. How do eco-evolutionary processes, this feedback between ecology and evolution, how does it shape the persistence of genetic and species diversity in a changing world? So this is the fundamental question that guides my research, and I primarily use both field and lab experiments using tools from both genetics and food web ecology to study these questions, or to answer this question. And a lot of the experimental work that I do, there's a feedback with theory where the work is strongly either developed by or influenced by theory, or I use theory to allow me to make some extra insights from my work. And now, while I'm fascinated with the food webs that I was showing you earlier, my work is really inspired by the evolutionary ecology of plants, insect herbivores, and their parasitoids, which make up much of Earth's eukaryotic biodiversity. So, and you'll learn more about the systems that I studied throughout my talk. Now, before we can start to answer this question, we first need to know, how does genetic variation within species scale up to influence the structure of food webs and the persistence of communities? And so for the next part of my talk, I'm going to show you the work that I've done to try to answer this question. So during my PhD at the University of British Columbia with Dr. Greg Crutzinger, I first became interested in addressing this question. And so Greg, he had established this common garden experiment that had consisted of 26 different locally collected willow genotypes of a common willow species in Northern California at Humboldt Bay National Wildlife Refuge. Now, this common garden was an excellent system to start studying these effects of genetic variation on food webs because willows host a really diverse community of insects and predators. But in particular, I became fascinated by this group of tiny flies that induce these galls on the plants. So these tiny flies, as larvae, when they feed on the plant, they induce the plant to growth to create these tumor-like growths called galls, which you can see here. And these galls not only allow them to feed on the plant, but they also provide protection from generalist predators like spiders, but they're still susceptible to being attacked by a specialized community of parasitoids, which you can see here on the top. And so one of the things that's really nice about this system is that it's relatively easy to build a quantitative food web like you see here. So here, each one of these lines, the width of the gray bars represent an interaction between two species, and the width of that bar represents the frequency of that interaction that I observed in this common garden. So the thing that I like about this system is that at the end of the growing season, I could collect the galls, bring them back to the lab, and rear them to determine whether or not the insects survived, and if they didn't survive, which parasitoid that they were attacked by. So for each of the different willow genotypes, I could build a quantitative food web. Now, in addition to building these quantitative food webs for each genotype, I also surveyed 40 different plant traits, including traits that determine the architecture of the plant, the toughness of the leaves, how hairy they are, but also the chemical diversity of the plant, to try to link genetic variation in the willows to these phenotypes that might influence these trophic interactions. And so what I found from this work is that different willow genotypes were associated with different sets of multi-trophic interactions. And so leveraging this data, I used rarefaction to infer how changes in genetic variation in the willow population would infer this property of the food web, the complexity of the food web. And what we found was that increasing willow genetic variation increased the complexity of the food web by as much as 20%. So I was really excited about these results because they're one of the first attempts to link genetic variation within species to the structure of interactions between species, which we know, kind of as I showed you earlier, has important consequences for the dynamics of populations and communities. But I also was aware that there was a number of limitations of this work. So for example, in this experiment, I didn't actually manipulate genetic diversity in the willows. I was just using a kind of a statistical technique, rarefaction, to infer the effects of genetic variation. Also, there wasn't really a strong link to the stability of the food web or the dynamics of the community. Because there's certainly some theory that suggests that more complex food webs are more resilient to environmental change, but I could also show you that some theory suggests that actually the opposite, that more complex food webs actually might be more fragile and less resilient to environmental change. So we didn't really have this strong link to the stability or persistence of the food web. Also, we had really no idea of the genetic details underlying the phenotypes that influence these trophic interactions. But what if we did? What if we knew the specific genes that were involved in these trophic interactions? I think this would have the potential to transform our understanding of how eco-evolutionary processes shape the persistence of genetic and species diversity. So to address these limitations, I developed a model food web during my postdoc at the University of Zurich in collaboration with my postdoc advisor, Jordi Visconti. At the base of this food web is the genetic model plant, Arabidopsis thaliana, and it's fed upon by two different species of aphids that are specialist herbivores on other plants in the Brassicaceae. And they're also attacked by this parasitic wasp, forming this diamond-shaped food web. Now, these insects, they're naturally associated with Arabidopsis, but the other nice thing about them is that they have relatively fast generation times, which enable me to study the dynamics of this community over time. And also, the availability of genetic resources for Arabidopsis really made this an excellent system to test the effects of genetic and phenotypic variation on species interactions and food web dynamics. And so one of the key phenotypes that we thought was important in mediating trophic interactions in this food web were these group of chemical compounds called aliphatic glucosinolates. Now, these chemical compounds, these glucosinolate compounds, you might not be familiar with by name, but I can assure you, you've definitely tasted these compounds. They're the same ones that give Brussels sprouts or cabbage its bitter taste, but also that spicy flavor to mustard and wasabi. Unfortunately, the genotype to phenotype map of these glucosinolate compounds in Arabidopsis has been really well studied. And so here, this is a simplified schematic of the biosynthetic pathway that determines much of the natural chemical variation in these aliphatic glucosinolate compounds. And so what you can see is that phenotypic variation in these glucosinolates is determined by 3 genes that function in a sequential pathway. So it's determined by MAM1, AOP2, and JSOH. And depending on whether these genes have functional or non-functional alleles determines the chemical phenotype of Arabidopsis. So just as an example, the Columbia genotype of Arabidopsis, which is one of the most well-studied natural accessions, it has a functional MAM1 gene, but a natural loss of function mutation at AOP2. So this Columbia genotype primarily accumulates this 4-MSO compound. So it's the chemical compound that dominates its phenotype. So after kind of identifying this as a nice system for being able to study the effects of genetic and phenotypic variation, I teamed up with Dan Klibenstein at UC Davis, who's an expert in studying the genotype to phenotype map of these glucosinolates. And he gave me 3 different transgenic lines of Arabidopsis that turn on and off these different genes, MAM1, AOP2, and JSOH, in a Columbia genetic background. So together with these 4 different genotypes, this provides a nice system to test the effects of specific genes on food webs, but also leveraging but also using genes that create much of the natural chemical diversity in Arabidopsis. And so together with these 4 different genotypes, I created experimental plant populations that had either 1, 2, or 4 different genotypes. And so with these 4 different, with this gradient of genetic diversity, this enabled me to not only test the effects of specific genes, but also test for a more general effect of genetic diversity on these species interactions. And so I grew these plants for 4 weeks in the greenhouse before adding my experimental food web. So here's a little video of me adding the food web. I've just added 2 individuals of one of the aphid species, and now I'm going over to add 2 individuals of this other aphid species to this pot of Arabidopsis. I then allowed these aphid populations to grow, to increase in their abundance for 2 weeks before adding the parasitoids to complete this food web. This just gives you also a bit of a scale for the size of the experiment that I was conducting. And so once a week, we would, I forgot to mention, we would put the cages in growth chambers, and once a week, we would pull out the growth chamber, or pull out each of the cages and count the abundance of each of the aphid species, which you can see, so Brevicorn brassica and Lipaphus erythemi. So you can see they're pretty easy to distinguish. But in addition to counting the aphids, we would also count the abundance of parasitoids, which we could see from the adults flying around the cage, but also we could identify them from these by looking for these brown mummies. So if you're not familiar with parasitoid biology, it's pretty gruesome. So an adult female wasp will lay an egg inside one of these aphids. This egg will eventually hatch and consume the aphid from the inside, and eventually, when it pupates, it turns the aphid into these brown mummies before it'll cut out a hole out of the mummy and then emerge and continue this process again. And so we repeated this process. And so I forgot to mention, after counting the insects, we would also replace plants to maintain resource availability throughout this experiment. And then we would repeat this procedure. We repeated this procedure for 4 months. Okay, every week, growing plants and counting insects. And so with this data in hand, we can now start to answer the question, how does genetic variation scale up to influence the structure of species interactions and the persistence of food webs? So I'm going to start by showing you the results that test for genetic effects on the overall rate of species extinctions, okay, from this experiment. In other words, food web persistence. So on the x-axis here, we have the week of the experiment, and on the y-axis, we have the overall survival probability. And what each one of these curves here represents the survival curve or the extinction curve for each level of genetic diversity in my experiment. And what we found was that on average, genetic diversity increased food web persistence, in that for every genotype that we added to the plant population, it increased the persistence of the food web by 19%. Now, we were really excited about this result, but it still didn't really give us information about the underlying genetic effects on these interactions. So we looked a little bit deeper to see whether which genes might be important in influencing this result. And this is what we found. So on the x-axis here, I have each of the different genes that I tested, and on the y-axis, we have the extinction rate, okay, this ratio of the extinction rate. And so what you can see here is that for both MAM1 and JSOH, depending on whether there was a functional or non-functional allele at these genes, it didn't really have a clear effect on food web persistence. But the story was different for AOP2. And in particular, what we found is that if we replace a functional AOP2 allele with a non-functional allele, that it decreased the persistence of the food web by 16%. Now, this 16% is actually a bit of an underestimate, because if we actually make the comparison of a functional allele to a non-functional allele, we saw that this allelic change increased food web persistence by 29%. So analogous to Bob Payne's experiment, where he showed that removing a species could have a strong effect on the persistence and in maintaining diversity of the food web, what we're finding is that AOP2 appears to be functioning as a keystone gene, such that depending on which allele is present in the plant population, it has a major effect on the persistence of the food web. So we were excited about this result, but it still didn't really give us information about, okay, now the question is, how exactly was AOP2 promoting food web persistence? And so to look at this, we looked at different transitions in the food web, which you can see here as in this graph. And so these food web transitions were local extinctions that simplified the food web over the course of the experiment. And so what we found was that, let me explain this figure a bit more. So each of these colored panels represents a different food web structure that we observed over the course of the experiment. And the arrows between two food webs indicate the food web transitions we observed. So solid arrows represent positive effects, and dashed arrows represent negative ones. And so what you can see here is that, and this is what we observed when we add a non-functional AOP2 allele to the plant population. And what you can see is that adding this null AOP2 allele, it had a specific effect on the food web, in that it actually prevented this dominant plant aphid parasitoid food chain from completely collapsing to their Arabidopsis only state. So that's where we see AOP2 primarily acting within this food web to promote food web persistence. it prevents the collapse of this food chain. So now we know where AOP2 is. Now we know where AOP2 is acting in the food web, but we still didn't really have a good kind of understanding of, okay, how exactly was it promoting persistence in this food chain? So to answer this question, I turned to ecological theory on structural stability. Now, for this theory, we need information on the interactions between species as well as their intrinsic growth rates. And I estimated this using this kind of Bayesian multivariate autoregressive model from the community time series data. Just as a bit of a kind of a broader perspective of what I did is taking advantage of all of the insect abundance data that I had. I used these data on the time series to infer what the food web structure actually was. And this is what we found. Okay, so on the x-axis here is the normalized intrinsic growth rate of the aphid, and on the y-axis is the normalized intrinsic growth rate of the parasitoid. This gray region shows you the combination of intrinsic growth rates of the aphid and the parasitoid where they're able to coexist, whereas this white area shows where they go extinct. And so what we can see here is that this effect of the non-functional AOP2 allele, it didn't alter this area where the aphid and parasitoid coexisted, but it actually increased the intrinsic growth rates of both the aphid and the parasitoid. So it moves from this region where they go extinct into this region where both the aphid and parasitoid are able to coexist. So now we know, okay, AOP2 promotes food web persistence by increasing the intrinsic growth rate of the aphid and the parasitoid. But why? How is it actually promoting their persistence? And what we think is happening is that this AOP2 allele, it doesn't just affect the chemical phenotype of Arabidopsis, but it also has strong pleiotropic effects on the growth rates of the plant. So these are results from a separate experiment, an additional experiment without any insects, where I repeated everything that I did before, just with no insects. And what we found is that plants with the null form of the AOP2 allele had a very strong effect on plant growth rate. So we think this positive effect of the null AOP2 allele on plant growth had this cascading effect. So it increased the intrinsic growth rate of the aphid and the parasitoid. And we think that this is the pathway that underlies this specific gene's effect on food web persistence. So just returning now to this first question, how does genetic variation scale up to influence food web structure and persistence? We did observe this positive effect of genetic diversity on persistence, but importantly, this positive effect was determined by a single allele at a single gene. And so I think this result has important implications because what it suggests is that the current loss of genetic diversity that we are witnessing could have abrupt effects on ecosystem persistence and functioning, because what it suggests is that some systems, they might be susceptible to a particular loss of a particular allele or a particular allele in that population. Now, you might be thinking, okay, Matt, these genetic effects, they're interesting, but surely in the context of environmental change, like climate change, these effects might be small. But what I didn't show you is that we replicated this experiment at 2 different temperatures to kind of simulate what the effects of increasing of warming would have on the food web. And what we found is this effects of this keystone gene were persistent across both food web treatments. And if anything, these effects were actually just as strong or stronger than the effects of warming on the food web. Okay, so far I've shown you how genetic variation can promote species diversity. Now I want to turn to the flip side, to start to explore how do changes in ecological communities, how could that feed back to drive evolutionary change within populations? And in particular, the question that became interested in answering was, how do extinctions from an ecological community, how are those extinctions going to shape genetic variation in remaining populations? And I think this is an important question to ask because we know that biodiversity is declining at an alarming rate, but I also don't think we have a good understanding of how changes in this community context, the loss of biodiversity at the community level, will affect the ability of species, the remaining species, to adapt to future environmental change. And so to answer this question, we're going to return back to the food web that I studied during my PhD, because this is also when I was interested in this question. But we're going to focus on one of the galling insects. Okay, so this leaf galling midge, Idiomyia silicisphaerica. And as you can see, it's attacked by 4 different species of parasitoids. These 3 parasitoids on the top are this guild of larval parasitoids. So they attack after the gall's already formed. And there's also this amazing little wasp right here. It attacks at the egg stage. So it's able to actually find this insect at the egg stage before the gall's even formed. Now, since I'm not as amazing as this tiny little wasp, I wasn't able to manipulate its presence or absence from this food web. But what I did was I conducted an experiment where I had allowed either some galls to be exposed to this natural food web, or I used these tiny mesh bags to exclude this guild of larval parasitoids. And then again, kind of like I did before, I would collect the galls, bring them back to the lab to measure their survival. But importantly, I also kept track of a few different traits that I thought a few different traits of the galls that I thought might be important in influencing their survival in the face of parasitoids. And so with this data and certain assumptions, I could start to make some inferences about how this guild of larval parasitoids would influence the evolutionary potential of these gall traits in the next generation. And so what we found was that this simulated extinction of this guild of larval parasitoids, it actually, in general, it acted to decrease the evolvability of this gall's traits in the next generation. Okay, it seemed to decrease the evolutionary potential of these traits. So what this suggests is that even though these consumers have a negative effect on the population abundance of this herbivore, that the extinction of these consumers could actually constrain the adaptive potential of this insect in the future. Now, again, I was really excited about this result because we're starting to see how changes in the structure of species interactions could affect genetic and phenotypic variation. But I was also still aware that it had its limitations. So, for example, this is a really coarse manipulation of food web structure. I was only able to exclude this whole guild of parasitoids, and I wasn't able to reciprocally kind of manipulate this guild as well as this guild, and wasn't able to manipulate the presence absence of certain species. But also, I only measured the effects of this food web on selection acting on this insect over a single generation. So right now, I still didn't have really good, solid information about how it would affect the evolution of this species over time. And so to address these limitations, I went back to the model food web. I made some modifications to the model food web that I developed during my postdoc to try to better answer this question. So one of the things you'll immediately see for this system is that I've added more species. So the species I added, one of them was this generalist aphid, the green peach aphid, Mysis persica. I also added this parasitoid, Ophidius colmani, which is a specialist on this aphid. And I also added a hyperparasitoid. Okay, so a parasitoid of the parasitoid. So adding another trophic level to this food web. Now, I studied how and I decided to focus on the evolution of this green peach aphid for a few different reasons. So one, I wanted to work with an aphid because aphids reproduce clonally during the summer. So what this allowed me to do is actually I could track evolutionary change by monitoring changes in the frequencies of the different aphid clones over the course of the experiment. Also, the green peach aphid is one of the most well-studied aphids, and there's already a reference genome available, which would make subsequent genetic analyses much easier. But the other thing, which I got really excited about when I was in the field collecting these insects, was that this aphid appears to display this red-green color polymorphism in Zurich, such that I was able to find one genotype that was a red morph, and the other 5 genotypes were green. And the reason why I got excited about this was because what this would enable me to do is track phenotypic evolution in real time simply by monitoring changes in the frequencies of these different color morphs over the course of the experiment. And I forgot to mention that I collected all of these insects from Zurich, from insects occurring on brassica plants around Zurich, just to kind of ensure a realistic evolutionary context. And so to study the evolution of this aphid species, I embedded it in a gradient of food web complexity. So in total, I made 12 different food web configurations, ranging from the aphid alone with the plant by itself to having up to 5 additional species. So I also had food webs with 1 additional species, either an aphid or a parasitoid, 2 additional species, or all 5 additional species, creating the most complex food web. Because this would enabled me to really kind of conduct a test of how changes, how the loss of species from this community might affect the evolution of this aphid. And so this is just a couple pictures of me setting up this experiment. As you can see, I used kind of the same cage design, and in total across these 12 different food web configurations, we had 60 different cages. And so for each one of these food webs, I established an initial population of the focal green peach aphid. And so right here, this is just a picture of like a tiny little petri dish where I added 2 individuals of each of the different clones. And so in total, I had 3 of these. I added 3 of these dishes to each of the cages. So I had 36 of these aphids in the initial founding population. And of course, as you can see here, if you're not colorblind, it's relatively easy to distinguish the red and the green morph. And so depending on the food web treatment, I had, this is just kind of gives you a timeline for the experiment. For different aphid competitors, I added them at the same time as the focal aphid at week 0. For treatments that had parasitoids, I added them at week 1 and again at week 2. And if the treatments had a hyperparasitoid, I added them at week 2 and again at week 3. And over the course of the experiment, I monitored multiple changes at different levels of organization. So I analyzed changes in the community or the composition of species in these food webs. I also tracked changes in the population dynamics of the focal aphid species. I also tracked changes in the relative frequency of the red morph to document phenotypic change in the population. And toward the end of the experiment, I used whole genome resequencing of pooled samples from each of these cages to reconstruct the genetic composition of these different aphid populations. And I did this at week 6, 8, and 10 of the experiment. And now, just for the purposes of this talk, I'm just going to focus on the changes we observed at the phenotypic and genetic level in this focal aphid species as it's embedded in this gradient of food web complexity. So this is what we found. On the x-axis here, we have each one of the different food web treatments. And on the y-axis, we have the trajectory of the red morph. Each one of these points represents an individual cage, and the red points, the larger red points, represent the average across those cages. So points above this dashed line simply indicate that the red morph increased in frequency over the course of the experiment. And so one of the things you'll immediately notice is that these 4 treatments on the right strongly favor the evolution of the red morph. And so one of the common things about these treatments is that they each have the letter A. You can see right down here, which means that each of these food webs had this specialist parasitoid, Ophidius colmani, which seemed to be really important in determining variation in this red-green color polymorphism. But these aren't the results that I'm most excited about. What I also want you to notice is that there's variation among these treatments in the trajectory of the red morph, and that's where we see the strongest effect of food web complexity. So on the x-axis here, we have the number of additional species that I added to the food web, and on the y-axis, we have the variation in the trajectory of the red morph, which is simply the standard deviation of the points that I showed you in this previous figure, for each treatment. And so what we see is that as we increase the complexity of the food web, it had a strong positive effect on the variability of the evolution of this red morph. Or in other words, increasing the complexity of the food web made the evolution of the red morph less predictable. But interestingly, we also observed this at the genetic level. So this is an ordination diagram representing the genetic composition of each one of these different populations. And for simplicity here, I'm just showing you the simplest food web represented by yellow points and the most complex food web represented by these purple dots. So dots that are further away from each other are more different in their genetic composition. And so what I want you to see here is that this shaded region created by these yellow points is much smaller compared to the shaded region of these purple points. And so what this again suggests, it kind of recapitulates the first result by suggesting that in more complex food webs, the outcome of evolution, the evolutionary trajectory of this aphid seems to be less predictable among the different cages. So kind of returning now to the second question, how do extinctions shape genetic variation in remaining populations? What we're seeing is that extinction from species in a community could actually act to erode genetic variation between remaining populations. And that so what this suggests is that this genetic homogenization among these different populations could constrain species potential to adapt to changing environments. So now just to kind of wrap things up, I want to return now to kind of the question that I started with. The main question that guides my work is, how do eco-evolutionary processes shape the persistence of genetic and species diversity in a changing world? So what my results are suggesting is that genetic variation can cascade up. It can have a positive influence on the persistence of species, which can promote species diversity. But also, since these extinctions from a community can feed back to affect genetic and phenotypic variation, what it's suggesting is that there's this positive feedback where genetic variation promotes species diversity, and that this species diversity promotes genetic variation among populations. And so this positive feedback is great for intact ecological communities, because what it suggests is that there's this engine for biodiversity where genetic and species diversity kind of promote each other in maintaining that diversity. But what it also suggests is that the loss of biodiversity at one level of organization, such as the loss of genetic diversity, could erode biodiversity at the other level of organization, which is worrying in the context of environmental change, because we're starting to lose biodiversity at these different levels, and it could almost promote further loss of biodiversity at each of these levels. And so, of course, this work I've done, I had highlighted some of my collaborators, but there's many other people and institutions that supported this work. And I just kind of want to quickly end by saying, at least for the grad students in the crowd, I know Gustavo had mentioned it. I've just started up my new lab at the University of Sherbrooke in Quebec, Canada. So if you're interested in any of this line of work and want to help build a more predictive gene to ecosystem ecology for a changing world, please get in touch. Thanks for your attention.
¿Preguntas?
Thanks for an interesting talk. At the beginning of your presentation, you said that there's this alternative theory where simpler networks can be more resilient
Yes.
to extinction. Can you speak a little bit more about that? Because that, I mean, your work shows the opposite, right? That the more genetic diversity, the more well resilient it may be, or the persistence is better.
Yeah. Okay. Yeah. So let me change. Well, I should have been a bit more careful with my words. Usually, that what most theory would focus on is like biodiversity at a particular level. So I guess I was talking about species diversity, kind of creating more complex food webs, and that those might be less resilient to environmental change. But I guess what we could see though is that if there is loss of species diversity, if there's still genetic diversity within a population, that could still promote diversity. Does that, or did you want me to speak to the particular theories, or kind of how that reconciles with my results?
Yeah, I was just wondering how they both come together, because you did mention this problem with climate change and losing species diversity.
Yes.
But I was wondering if there's a scenario where you could have a few resilient species that could survive the catastrophe and then promote divergence.
I think so. Yeah, I do want to kind of, I guess, give a bit more hopeful message, because I think you're totally right. Because we might also see that operating in more kind of temperate ecosystems, where kind of I'm from, where they might be dominated by a certain species. But if there's lots of genetic variation within that species, even though there might not be a ton of diversity like among species in the community, that could still foster, I think, biodiversity. So does that kind of answer the question? I think genetic diversity can sometimes compensate. Yeah, compensate for
that. Thank you, Dr. Barbour. I would ask you that this is a very attractive view or scope for, let's say, quantitative population biology, and it's related to climate change, but I can help it to think about crops.
Yeah.
So do you think that this kind of theory or developments can help in deciding something about biological control, especially the kind of species that can be introduced to control some kind of parasite, phytoparasite?
Yeah, so I don't have like this probably isn't the best slide for that, but I have thought about that. Because one of the things, it makes me think about two different things. So one of the things that we could do in terms of biological control is that if we increase like genetic diversity, like let's say of a species that, let's say a parasitoid that we want to help control a particular herbivore, if we inoculate that population with genetic diversity, it actually might make it gives it more potential for it to adapt to that herbivore that it's trying to suppress. So in that way, it could be beneficial. The other thing that I think is really interesting is that in the context of crops, I think a lot of work has done a fantastic job of trying to like understand genes that influence direct interactions with other species like a herbivore. But the thing is, I mean, in these crop ecosystems, you look and that there's lots of species that are actually attacking these plants, and they're embedded like within a food web. So I think the intersection I see there is that if we start to think about these indirect effects that could be caused by these genetic changes, maybe it would give us a better perspective on how these communities, we might be able to build more resilient kind of crop systems that can also kind of act to promote biodiversity at the same time. Does that kind of answer your question? I think thinking more about these indirect genetic effects would be helpful. and accounting for the fact that in these crop ecosystems, it's not just one species, but it is part of a food web.
Thank you.
¿Alguna otra pregunta? I have one.
Yes.
So regarding this, the increase in the population of the red aphid, once you have one particular parasitoid.
Yes.
So that was resulting from that parasitoid eliminating or infecting more the green aphid than the red one?
Yes.
And do you know what at genetic level will determine that, or
no?
If it is...
Yeah. So yeah. Again, I just want to point out that that's the frequency of the red morph. So it's not like it increased. It actually that specialist parasitoid caused the aphids to go extinct. If it was there by itself, it would eventually cause the aphids to go extinct. But the red morph would increase in frequency. So it was more resilient. It was more resistant to the parasitoid. But so that's kind of one question. But the genetic details, we really, I think I can't get at right now because I only use 6 genotypes. For that, I think we would need a much broader array of genotypes to start getting at that. Because what I fully acknowledge is that it might not necessarily be the red morph, the color variation that's under selection. It could be some other chemical phenotype or some other, maybe it has a particular endosymbiont that actually makes it more resistant to the parasitoid. So the red morph was basically a marker for tracking evolutionary change, but we don't know those genetic details. It'd be interesting to look at that.
The other thing I'm wondering is in change seasons, normal year seasons, where the plants, depending on the amount of energy that receive the sun, change the production of these chemicals that determine whether insects like them or not.
Yes.
Is it? What about that? Can you observe changes in populations of the insects and parasitoids with seasons and during the year, or?
Yeah, I think that would also be like another very interesting direction to go, because right now I do recognize a lot of this it's within kind of a lab. I'm kind of creating these lab ecosystems, but that there also might be definitely ontogenetic changes over the course of the season. Like I know oak trees, which are around here everywhere. At the beginning of the season, their leaves are a bit tastier to herbivores, whereas at the end of the season, they develop more tannins before they senesce, so they're less susceptible. So I think accounting for that variation would be interesting. I haven't done it here, but I think you could start to make predictions about how the chemical phenotype of the plant could influence these interactions, but I don't know if it would act synergistically or kind of additively.
Omar, ¿hay preguntas en YouTube? ¿No? ¿Alguna otra pregunta? Bueno.
Hi, thank you very much for your presentation. For me, as a microbiologist, I was wondering, how can we integrate the genetic diversity in microbes into your study? And if it's possible, how to do so?
Yeah. So I guess I would say I haven't thought too much about genetic variation within like microbial species, but I tend to think of, even though we use genetic techniques to like maybe infer the composition of these microbial populations, I guess what I would say is that it's much harder to do, but just recognizing that within a microbiome, it is a network of interactions. And maybe if we can start to infer that network of interactions, it could help us be able to predict these phenotypic changes that would be more relevant. So it kind of doesn't quite get at the genetic variation within microbes, but it still speaks to that diversity. Does that kind of
help? At the level of species diversity, and how it translates, or how it affects the chemical phenotype of the plant, maybe because plants have interactions with bacteria.
Certainly, I know. I don't include the the leaf microbiome, for example, or the root microbiome, even though I think that would be super interesting to do. Fortunately, there are some of the collaborators at the University of Sherbrooke that are focused on that are microbiologists and also are studying the microbiome on the leaf and the root. So it would be interesting to collaborate with them. But
thank you very much.
It's a cool idea.
Sorry, I will extend a question on that, because also looking at your ideas over there in the screen, let's think about pollinators also. Can you elaborate, or can you mention some studies that have helped in the way that we can conserve, we can predict the displacement or the reintroduction of pollinators, especially because for very big reforestation efforts or things like that, it's a key component.
Certainly, yeah. So I don't know how good I'll be able to answer that question, but I guess what I will say is that, like, we know maybe the introduction of like certain pollinators, maybe like honeybees, you know, that could also, you know, it can maybe have an initial like positive effect from like promoting like actually promoting pollination services, but then it also might erode the ability of native pollinators, erode resilience at that level. So I don't know. I guess in that context, that makes me like think it's definitely a more nuanced, like a kind of a complicated picture. But I think I guess the thing that I would think about is just we just got to, and I think we know about this. Like we know that adding populations, we're going to add them into a community context where there's these interactions. I think as the community ecologists need to do a better job of actually really characterizing the networks of interactions that matter, so we could start to make these predictions. But I think that's super important to do.
Very much.
Thanks.